So this project is about string chemistries. So a brief overview on string chemistry, and then we can get into like what the actual project is. So string chemistry networks are a type of artificial chemistry networks where instead of trying to model real chemistry we see in real life, we make some simple model that has some like abstract simplified notion of what a chemical is and how chemicals can react. And these are often used to study chemistry in this sort of like zoomed out abstracted way to see if we can figure out what sort of like high level properties of real chemistry give rise to various phenomena we see in real chemical networks. So in our case, we're using string chemistry networks where instead of like real chemicals, we just have strings of characters that are joining and splitting. And we use these string chemistry networks to designate some arbitrary input and output metabolites on the networks and try to identify the minimal network of reactions that you need to convert your arbitrary input chemicals to your arbitrary output chemicals. And we thought this might compare to an evolutionary scenario where you have an organism that has certain metabolites in its environment that it can use as food, and it has certain metabolites it needs to make for itself to produce more biomass to grow. And you might see a reductive evolutionary process here trying to minimize the number of reactions that organism is using to produce biomass from its available food sources. So we thought we might simulate this under string chemistry networks and see, are there any sort of common features between these minimal networks you need to convert arbitrary inputs to arbitrary outputs. And interestingly, we did find that your choice of output metabolites for these artificial networks has a much stronger influence over what the minimal network in between input metabolites and output metabolites is, regardless of what your input metabolites are. So the choice of output metabolites or biomass precursors, to go back to our artificial organism analogy, is much more important for understanding the metabolic network of the organism than the identities of the food sources that organism is using, which is somewhat unexpected and may be relevant for how we go about reconstructing real metabolic networks, because it suggests that perhaps it is more important for us to thoroughly characterize the biomass compositions of all the organisms we wish to create metabolic networks for than it is to enumerate all of the food sources different organisms can use. So that's something. All right, so we got an overview of the project. Let's go into a bit more detail about what these string chemistry networks are. So we can see here at the bottom of this top left box, a very simple string chemistry network where we have where we have 6 different strings that can react in 4 different reactions. So our strings are A and B, and then pairs of A and B. And then the only reactions we allow in our network are reactions that either split one string into 2 strings or combine 2 strings end to end into 1 string. So we do not have any branching strings. These are all simple linear polymers. And the monomers here kind of represent atoms, basically. They have no internal structure, unlike most metabolites in a real network. But you could sort of also think of them as functional groups or in real chemistry networks. So this is clearly very simple. There are many more kinds of reactions that can happen in real life. But we went with this just to see, like, what does this simple model tell us about real chemistry? Is it comparable to real chemistry in some way? So one thing to note about these networks is that the size of these networks is completely governed by 2 things. How many different kinds of monomers or characters are you allowing? So in the case of this network we see here, there are 2 kinds of characters, A and B. And how long do you allow strings to be? So here, this is all the possible chemicals and reactions you get when you have 2 kinds of characters, and you can have strings of up to length 2. So 6 metabolites, 4 reactions, not many. But as you see in the top right panel here, this number grows very fast. So if we're looking at the number of metabolites, we have that in terms of A, the size of your alphabet, the number of different monomers you have. and L, the maximum length a chemical can have. So we have some functions here defining the number of metabolites, the number of reactions as you vary these parameters, and then these left two plots here plot those functions for several different values in A and L. And as you can see, the y-axes on these plots are log scales, so these are almost exponential growth, which is quite fast. So it doesn't take particularly many types of monomer or long strings to get in the realm of real metabolic networks in terms of number of metabolites, number of reactions. You can see here the numbers of metabolites and reactions in E. coli, yeast, and human metabolic networks. So that's interesting that they can get to the same size, but just because they're the same size, does that mean they're like, you know, structurally similar to real metabolic networks in any way? So one way we decided to look at that was look at the degree distributions of the metabolites in these string chemistry networks. And if you compare the degree distribution of the string chemistry networks to the degree distributions in E. coli and yeast, you see that they have the same general shape in this third plot here in the top right panel. So that's interesting. Suggests that at some abstract level, our string chemistry networks are structured in ways reminiscent of real metabolic networks. Furthermore, people often study real metabolic networks using flux balance analysis to assign fluxes to all reactions in the network. And all flux balance analysis FBA needs to do that is a stoichiometric matrix of the network and some reaction in the network that you're maximizing flux through or minimizing flux through, some constraint like that. And the string chemistry networks, we can certainly define stoichiometric matrices for them because you know they have reactions with reactants and products. We put coefficients on them. We got a stoichiometric matrix. FBA does not care that they do not represent actual chemistry. So we can make up some arbitrary biomass reactions here that are objective functions that absorb random metabolites as a sink and don't produce anything, maximize flux through those, allow certain random metabolites in our string chemistry network to be absorbed from the environment. And we can perform FBA on our string chemistry networks. So if we do that for a bunch of randomized choices of networks and what the input and output metabolites are, we can generate flux distributions through all these networks. And we can see that this distribution of fluxes, again, follows the distributions of fluxes you see in real chemistry network, real metabolic networks, which is, again, somewhat surprising. These are much simpler than real metabolic networks in many ways. So interesting that we see that. So back to this notion of, or going deeper into this notion of applying FBA to these string chemistry networks. If we look at the bottom left panel here, we see the string chemistry network with all possible reactions for chemicals that have two kinds of monomers and are up to length 3. So you can see this is already much more complicated than the one right above it, which is all possible reactions for chemicals of two kinds of monomers up to length 2. So we've applied FBA to this network, and we have arbitrarily designated this metabolite BAA at the top left here as a metabolite in the environment. And at the bottom here, you can see our little pseudo biomass reaction that takes 3 random networks from the metabolite and turns them into nothing. So we have set that biomass reaction at the bottom as the objective function, maximize flux through it, and assigned fluxes to all the reactions. And all these reactions with black arrows are reactions that have flux. And the green arrows are sort of input and output fluxes. So at the top, we have a green arrow taking our one food source in. At the bottom, we have green arrows taking our biomass precursors to our biomass reaction to, you know, throw them into the void. And then we see all of these gray arrows are reactions that FBA did not assign a flux to, did not decide were necessary for maximizing flux from this food source to this biomass precursor. So we looked at this and we thought, so surely we have not assigned fluxes to many reactions in this network, but could we use fewer reactions in this network to still accomplish the same metabolic task of turning this one food source into these 3 biomass precursors? So we devised what we call the pruning algorithm. We take a universal network of all reactions. We do FBA on it to assign fluxes to all of the reactions in this network. We remove all the reactions that FBA did not assign a flux to. We find the remaining reaction that has the smallest assigned flux, delete it, do FBA again, and see, can we still accomplish our metabolic task? Can we still produce our biomass precursors from our food source? If yes, we repeat this process until we cannot delete a network and a reaction from the network. And once we can no longer delete anything, the network is pruned. We have a pruned network. So we repeated this process for a bunch of different networks with a bunch of different food sources and biomass precursors, and then set out to see, all right, what can we tell about the structures of these pruned networks? Is there anything that can predict for us which reactions get left behind in the pruning process? So we stepped it up a bit to the universal network of reactions and metabolites, where there are 2 kinds of characters strings can be made of, and strings can be up to 5 characters long. And we aren't visualizing that network here because it is a mess. It is much more complicated than the one you see in the bottom left, which is only up to string length 3. So we made a bunch of copies of this universal network, and then each of these universal networks got 2 input metabolites from a set of 100 input metabolites, so a set of 100 food sources, nutrient environments, and then 5 biomass precursors, so from another set of 100 different biomass reactions. So we have every possible combination of these 100 pair environments and these 100 5-metabolite biomass reactions. And we pruned all of them and wanted to compare the structures of these pruned networks. So in order to visualize these pruned networks, because they're all complicated and messy, and it was hard to like align the networks themselves to visually inspect them, we represent each pruned network as a binary vector with where we have all the reactions in a specific order for the universal network. We have a 0 if it's not present in the pruned network we're looking at, and a 1 if it is kept in the pruned network we're looking at. So we can represent each of these, 10,000 pruned reactions or pruned networks as a binary vector of which reactions are in those networks. And then we can use UMAP to visualize all of these different binary vectors and have a little point representing where each pruned network is. And so those are the UMAP scatter plots you see on the right here. And the first thing we checked was, all right, so 2 things we know about each pruned network. We know what the biomass reaction was when doing FBA to prune the network, the objective function. And we know what food sources that network could use to make those biomass precursors. So in the top plot, we colored each network by which of the 100 biomass reactions it had. And in the bottom plot, we colored each network by which of the 100 2 nutrient environments the network had. And as you can see, there's actually some light clustering by colors going on in the top plot and not at all really in the bottom plot. So this tells us that the structures of pruned networks that have the same biomass reaction are much more likely to be similar than the structures of pruned networks that happen to have the same environment but may not have the same biomass reaction, which is not what we expected, somewhat surprising. It's not immediately clear that one of those would have much more influence over the structure of the network than the other, but here we are. So we did a number of other analyses of these string chemistry networks, not shown here because it's a poster limited space. So if you want to read more about what we did with these string chemistry networks and the many properties you can investigate about them, The title of this poster up at the top is a hyperlink to a paper we just published in the Journal of Molecular Evolution, maybe a month ago. And if you want to look at the software we use to generate all of these visualizations and make all of our string chemistry networks and do this pruning algorithm, the pruning algorithm title on the little box there in the bottom middle takes you to our GitHub repository where we have all the software we use to make this and visualize these networks and make all of these figures. So I hope you take a look at our papers and enjoy the string chemistry network. And feel free to interact with the poster webpage here on the ISMB website if you have any questions or comments. Thanks.